we are going to take a moment and we're going to kind of pivot back and talk more about cells. So we got really, really zoomed in last unit, looking inside of organelles. We're going to kind of zoom a little bit back out, and we're looking at cells as a whole. And we're going to look here at mitosis, which is cellular division, right? And so here we go. We have an original cell, and then that cell is going to replicate its DNA, and then it's going to split into two cells that have the same makeup as that original parent cell. Okay, these two cells that come about are called daughter cells. So we're going to be looking at this process and specifically focusing on the genetic material, the DNA, and how it moves through this process. An important thing to note is that these are how your body cells divide, okay? And a sciencey word for body is somatic. Okay, so this is how your somatic cells divide. This is not reproductive cells. This is not sperm and eggs. That's a different but similar process. We'll learn about that later. This is your body cells, skin cells, liver cells, stomach cells, kidney cells, what have you. Okay, and so we want each cell to have a complete, like all of the DNA it's supposed to have. That should make sense, right? It doesn't want to be missing any instructions. Okay, and when you look inside of a cell with its DNA, it has 23 chromosomes from mom and 23 chromosomes from dad. So we have two sets of 23 chromosomes. That's what makes a complete body cell. So up here, obviously this isn't drawn proportionally, but this would have two sets of 23. And after mitosis, this would have two sets of 23, and this would have two sets of 23. Okay. So we have 23 pairs of chromosomes, aka like 46. That's 23 times 2. All right. So obviously chromosomes are going to be important. So let's talk more about them and get into some vocab. Okay. Now, if we take DNA, just regular old DNA, right? The double helix, that should be familiar. But as it turns out, if DNA was just like this, it could get tangled really easily. Think about like string. Think about how easily string gets tangled, which is why like yarn, you ball it up. Okay, so basically that's what DNA does. So if we wrapped DNA around these little proteins called histones, as soon as that DNA is wrapped around these histones, now of course we give it a new name, and that new name is chromatin. It's still just DNA, but it's DNA like wrapped. Okay, and then that chromatin is going to start coiling up even more, like a cord on an old school phone. You know what I mean? Okay. That's going to coil up, and the coils are going to fold back and forth, and this thing is a chromosome. And so that's when the chromatin is condensed, aka coiled up. So it's all just DNA, but a chromosome is like super coiled DNA, and a chromatin is, hey, now we're wrapped around some histones. Then that part is going to coil even more, if that makes sense. Okay. When we're doing mitosis, it's going to be important for your chromosomes in your DNA to be coiled, so they don't get tangled and they can get moved around. Okay, so DNA will be in its chromosome form during mitosis. Okay, but when mitosis is done, DNA is going to unravel again back into chromatin, and it's going to hang out in chromatin until it's time for the next cell division. Okay, so DNA only coils up into chromosomes during mitosis. Here's just another graphic to show you. Here's a double helix wrapping around histones, right? That is chromatin. And now the chromatin coils and coils and coils. And here is a chromosome. Some more chromosome vocab. This would be a chromosome. If that DNA gets replicated, this would be a duplicated chromosome. Okay. This little part here in the middle where it comes together, this part is called the centromere. This left side and this right side are identical. And so the left side and the right side, they're called sister chromatids. So the left side would be a chromatid and the right side would be a chromatid. Now, here's the crazy thing. This is all a chromosome. This is one chromosome and this is one chromosome. The difference is this is an unduplicated chromosome and this is a duplicated chromosome. But we still count them as one because they're connected right here in the middle of the centromere. One chromosome, but this chromosome has two sister chromatids. Blah, I know, vocab. Okay, so before cell division, the cell's in a phase called interphase. And basically, this is where the cell's just like living its life. It's doing its thing. It's doing all the reactions it needs. It's growing. And the cell's going to replicate its organelles. Because if you're getting ready to divide into 2 cells, you're going to need double the organelles so that each cell gets a full set of organelles, if that makes sense, okay? So the mitochondria, the ER, the Golgi, all that stuff are going to get replicated. And also the DNA is going to get replicated too. Again, if you want each daughter cell to have a full set of everything, you got to make a copy of everything first. So all the organelles get copied, all the DNA gets copied, okay? And then your cell is going to get a signal to divide, which we're going to learn more on that later. And so once it gets that signal to divide, then we're going to move into the actual process of mitosis, which happens in 4 phases. The phases of mitosis are prophase, metaphase, anaphase, telophase. I think of it as PMAT. If you want to come up with a different thing to remember that, you do you. Okay, so this is mitosis. Okay, so mitosis really specifically is dividing the genetic material. And we'll talk at the end, cytokinesis, when the 2 cells actually separate, is not a part of mitosis. Notice how this blue arrow is not a part of the red mitosis arrow. So again, we'll talk about that later. So let's go through our 4 phases of mitosis. First one up puts the P in PMAT, prophase. So here is where our DNA condenses down into chromosomes. So that means they're going to become visible, okay? When the cell's in interphase, you can't see chromosomes. But now all of a sudden, we're going to start being able to see our chromosomes. The nuclear membrane or the nuclear envelope is going to start breaking down because we're going to need to move these chromosomes around, and we can't if they're enclosed. Okay, we're going to have these things called centrioles right here, and they're going to start moving towards opposite ends of the cell. And these little strings right here are called spindle fibers, and they're going to start forming. They're going to grow out of the centrioles. These little tubes are the centrioles. Okay. And so slowly these spindle fibers are going to start to find the chromosomes, and they're going to start to attach to them in the middle of the centromere. Okay, and this is the longest phase of mitosis. Here's an example of a cell in prophase, this like dark blue color. This is the DNA. So here we're seeing the chromosomes starting to get kind of clumpy. That's a technical term, clumpy. Next, PMAT, metaphase. And so now we've got full attachment of our spindle fibers to our chromosomes. Look at our spindles connected at the centromere. Here's our centriole spindle fibers, right? And so those spindle fibers are going to push and pull and line up those chromosomes right down the middle of the cell. And so here's a picture of it, right? So here is our blue DNA lined up down the middle, and the green you can see are the spindle fibers there. Okay, there would be centrioles. You can kind of almost see one right there on either end of the cell. And then we've got anaphase. And so here our cell's going to go from being like a circle shape to an oval shape. And then our sister chromatids, aka our duplicated chromosomes, are going to be pulled apart, okay? And one sister chromatid is going to go one way, and the other one's going to go the other way, and they're going to be pulled to opposite ends of the cell. This is the fastest phase of mitosis, okay? And so we're going to have things be pulled to either end. Next, we've got telophase. So this is our last stage of mitosis. And so for this, we're going to see our nuclear envelopes or nuclear membranes starting to reform. Okay, we're going to have our DNA kind of clustered on either side. You can see some cell membranes starting to pinch in the middle. Our chromosomes are going to begin unraveling back into chromatin form, and our spindle fibers are going to break down. So here's a picture of it. So you can see there's the blue, right? There's the blue over there. Okay, you can see that starting to pinch in like that. Okay. Now, after telophase, but it's not technically a part of mitosis. Okay, this is actually, once we have 2 separate nuclei, mitosis is technically done. But notice, do we have 2 separate cells? No, we don't. So the final thing a cell needs to do is called cytokinesis. Again, not technically part of mitosis. And this is where we actually have the actual final cell division. We have separate cytoplasm, separate organelles. We now have 2 separate cells. In an animal cell, the membrane just pinches in continually. So you can see this one is almost done here, right? The blue is the DNA that's unraveled in the nuclei. And notice the green, oh, look at that, almost done. In plant cells, though, here's a plant cell, and notice this dark line right here. That's the new, it's called a cell plate, but essentially that's where the new cell wall is growing. So with cytokinesis in plant cells, that's not complete until you got a cell wall going across the entire side, okay? But with animal cells, they just got to keep pinching in their membranes, okay? So again, important things would be that mitosis produces two genetically identical daughter cells from one parent cell. We would do this to grow, repair. If you're a single-celled organism, this might be how you actually reproduce your cells. That's called asexual reproduction. Okay, so this is how like bacteria reproduce. And again, here are the main highlights of what's going to go on in each phase. I'm not going to go and read them off. Okay, but there they are. And so for you, here are your things to consider, things you should know. And here's where we are in the textbook. Highly recommend that summary. It's really, really great.